Hello friends, I am Mayantika and in this video I will be discussing about asexual reproduction of a brown algae Ectocarpus. Earlier I have made videos on the morphology and ecological importance of Ectocarpus. Link I have put in the description box, and in this video I will be discussing about the sexual reproduction, and in the part 3 of the Ectocarpus video will be the sexual reproduction, and later it will be how Ectocarpus is used as a model organism in various branches of research. So this Ectocarpus, this brown algae Ectocarpus belonging to Isogeneratae class, unlike other Isogeneratae like Dictyota, Padina, Zonaria, this Ectocarpus is not belonging to the warm climate. It belongs to the temperate and polar region. And other class Isogeneratae algae like Dictyota, Padina, Zonaria, these belong to the warm climate. So the temperature, alternation of temperature alters the reproductive cycle in Ectocarpus. That is why I am mentioning the temperature conditions, because under warm condition it reaches maturity, reproductive maturity faster than when raised under its normal temperate climate. So overview of Ectocarpus. Now, as I was mentioning earlier, that the life cycle of Ectocarpus depend upon it is temperature dependent. Now, any Ectocarpus, it has two type of sporangia, especially Ectocarpus siliculosus, which is the most common species in the lab, common species of Ectocarpus that is been studied for various purposes, which is a model organism for various type of lab researches. So that species of Ectocarpus, it produces two type of sporangia, unilocular and plurilocular sporangia. But other species of Ectocarpus, it can produce one or either one of the type of sporangia, either unilocular or plurilocular. Now, this unilocular sporangia, it produces haploid Zeus pores. But on the other hand, plurilocular sporangia, it produces diploid Zeus pores. So the haploid Zeus pores produced by the unilocular sporangia at 13 degrees C, unilocular sporangia is produced, and at higher temperature, that is 19 degree C, plurilocular sporangia is formed. So we can infer that at higher temperature, the more sporophytic generation is increased, and at lower temperature we see more of gametophytic generation. But at intermediate temperature of 16 degrees C, at this of 16 degrees C, we see that both type of sporangia is developed. This is the case, that is the both type of sporangia develops. It is the case of only those species which produces both type of sporangia. For example, Ectocarpus siliculosus. So here we see that the tissue development is altered by alternation of temperature. So this sporophyte generation produces two types of sporangia, unilocular and plurilocular, and its production depend upon temperature. And this sporophyte in turn is produced by two processes. One is that it is produced parthenogenetically, when any gamete any gamete which does not it is not able to meet its opposite gamete, then it develops into a haploid plant, and later increases its ploidy number and converts to diploid sporophyte. And another is the normal process, that is by which the two opposite spores, they develop into a zygote, and this zygote later this two spores, they fertilize and develop into a zygote, and this zygote it germinates to produce a sporophyte. So sporophyte is produced parthenogenetically as well as by normal process of fusion and zygote formation and germination of zygote. And this sporophyte produces two types of sporangia. One is the elongated cone-shaped plurilocular sporangia. Another is the circular unilocular sporangia. Here we see that the circular unilocular sporangia that is borne on the tips of lateral branchlets, and here we see the plurilocular sporangia. It is borne, plurilocular sporangia is borne on the terminal tip of lateral branches. Here it is the tip of lateral branches, and here it is in the unilocular, they are borne on tip of branchlets but not lateral branches. So the difference between plurilocular and unilocular sporangia is that plurilocular, it is multicellular, and unilocular sporangia, it is unilocular. It is unicellular. So unicellular unilocular sporangia, it is produced at the tip of branchlet, and the spores that is developed is haploid. And the plurilocular sporangia, it is developed from it contains many cells, and it is developed from the terminal cell of branches, and the spore it produces is diploid spores. So plurilocular produces mitozoos, and the unilocular produces. So this is how the development occurs. The terminal cell of any lateral branches of the it develops into a sporangial mother cell. Sporangial mother cell, how it develops? Any terminal cell, it enlarges in size, it increases the number of chromatophore, and then undergoes a series of transverse division, and then a series of vertical division. So in this way, the single mother sporangia give rise to a plurilocular sporangia. So divisions, as the divisions is mitotic, so there will be no reduction in the ploidy level, and that is, it will be diploid in nature, and the Zeus pores that are biflagellate are diploid in number, they are called diploid in ploidy, they are called mitozoos pores. So mitozoos pores are produced by the plurilocular sporangia, are liberated by dehiscence of the apical aperture. There is this apical aperture, this is the plurilocular sporangia, and the apical aperture, it dehisces, and the spores are liberated, and they swim for certain time before settling down to produce another sporophytic plant. So plurilocular sporangia producing diploid zoospore is just a mechanism to increase the number of sporophytes. And all the sporophytes, they are the clonal, they are just clonal clones of the mother plant. And about the zoospores, both the unilocular and plurilocular sporangia, that they produce a similar zoospore so far the morphology is concerned. So far the structure is concerned, all the zoospores, they are pyriform or pear shaped, and they have two flagella, that is biflagellate, and the flagella, they are inserted laterally and of unequal length. Here we see, this is the diagram, and the pear shaped pyriform the zoospore has two flagella. The forward is the tinsel type, which has a flimmer of hairs. This is the flagella with flimmer of hair. This is the flagella. This is the tinsel type of flagella with flimmer of hair in the front forward, and this tinsel flagella help in the movement of the Zeus pore. On the other hand, another, that is the whiplash type, that is directed backward, it is just dragged on and it rarely feeds. So this morphology of flagella, it is common for both the haploid myojoospore and diploid mitojoospore. So apart from that, this structure of zoospore, it contains some chromatophore and an eye spot. Eye spot is very close to where the flagella are inserted, or the flagella originate. The eye spot is mostly for photoreception, and the eye spot, it helps in photoreception, but otherwise the both the unilocular and so that was all about Zeus pore. Now, once the Zeus pore is liberated, it swims for certain time. Then what happens is that it puts in the anterior region. It settles down with the anterior region in the downward side, and it retracts the flagella. And once it retracts the flagella, a membrane is secreted, and once the membrane is secreted, then it rounds off and then germinates by putting forward a tubular projection. So if it is a mitozoospore, then this sporophytic plant is produced, that are clones of the mother plant. And but if myojoospore, that is a haploid zoospore, is produced, gametophytic generation will be produced. So, but whether it is sporophytic or gametophytic, one thing is that their tolerance level will depend upon the salinity. The sporophytes, they are more tolerant to the salinity than the gametophyte. This is how unilocular sporangia develops. Here we see in this A diagram, in the A diagram, we see that a terminal cell of a branchlet, it enlarges in size, it undergoes meiosis, that is when one 2n is converted, 2n converted to n. That is, this is a reduction division, and here one cell is converted to 4 cell, that is one diploid cell converted to 4 haploid cell. This 4 haploid cell undergoes repeated division. Here in C, it undergoes repeated division. So there is karyokinesis but no cell wall formation. So they divide repeatedly, and what they do is they accumulate protoplasm around it, but later they develop a membrane and they metamorphose into Zeus pores, Zeus pores. These Zeus pores, they are haploid Zeus pores, and as this mitosis has happened in the step B, so this Zeus pore, it is not all the spores are of similar type. The genetic material, it contains two type of chromosomes, that is one is U chromosome, another is V chromosome. So depending upon the gamete, they segregate. One single mitozoospore, one single myojoospore will not contain both U and V. So a single myojoospore will contain either V type or U type of chromosome in their spore. So this is how this, say for example, this is the U and this is the V containing myojoospore. So they develop into a gametophytic plant, Ectocarpus plant, and they both type, they produce different type of gametes. They produce different type of gametes. Gametes is also different. So these gamete, they finally they fuse, they form zygote, and this zygote they form again the sporophytic plant. So this is how the unilocular up to the stage F. Up to this stage, it is how the gametophytic plant is produced, and they produce gamete. So this will be up to this up to actually up to stage E. So this was all about what I was talking, that half of the Zeus pores, they develop into U and V type of chromosomes, and the V is for the male gametophyte, that is the male gametophyte, and the U is for the female gametophyte. So this was all about the asexual reproduction of Ectocarpus. In the next video, I will be making about the sexual reproduction of Ectocarpus, and also the previous video I made about the morphology and ecological significance of Ectocarpus. So this is all about this, and if you want me to make any videos regarding any other topic, do write it in the comment section, and please don't forget to like and share my videos, and also to subscribe. Your subscription will encourage me to make more such videos. Thank you for watching my videos.